You used to call me on my. You used to.
Hey Emily, what's up?
Hey, nothing much. Just waiting for it to be time to go to our lockers. Oh my gosh, you would not believe what just happened.
What?
Busted Jeeper just liked one of my posts.
Oh my gosh, really?
Yeah, let's take a selfie. Hey, look at this. The lighting here makes my eyes look different. Like two different colors. Imagine if my eyes were blue and green and brown.
Actually, Emily, when a person's irises are two different colors, it's called heterochromia. And heterochromia is a genetic mutation.
Really? What's a mutation?
2 hours later.
Okay, kids, take out your books. We're going to learn about genetic mutation today.
Okay, kids, so I'll be giving you the baby version of genetic mutation. You'll learn all the juicy details in molecular biology. So just stop me if you have any questions. You know I'll answer it because I'm the best teacher ever. Okay, so first we're going to discuss where mutations are found. So DNA is made into RNA, which becomes protein. This is called transcription. And this is called translation. Okay, so a mistake during translation would only affect one protein for each mistake. So since a mistake during translation would only affect one protein for each mistake, we can conclude that mutation does not arise at the protein level. Now, a mistake during transcription is also not considered a genetic mutation because it would only affect a few proteins for each mistake. So again, we can conclude that mutation does not arise at the RNA level. Now kids, if there is a mistake made at the DNA level, it is considered a genetic mutation because it would affect all later proteins that are made. Are you guys okay so far? Any questions?
So where do these genetic mutations come from?
Good question. That actually leads right into the next part of the lesson. Okay, so there are two ways to acquire a genetic mutation. The first way is through inheritance. Okay, so if a male and a female, fall in love and they have a child, and one of them, we'll say it's the guy, has genetic mutation, there is a chance that one of the children, or any of the children, can acquire the genetic mutation. This is the child that got it. Okay, kids, so you may be wondering, how does this happen? So, if the mutated DNA is found in the sex cells of one of the parents, in this case, the dad, there's a chance that the mutation will be passed down to the offspring, in this case, the little boy. So the other way that a mutation can occur is spontaneously. Okay, so a spontaneous mutation is when an organism acquires a mutation without either of the parents having it. The factors that cause spontaneous mutations can include environmental factors such as UV radiation or environmental poisons. Or it can just occur randomly. The two ways that you can acquire genetic mutations. Again, inheritance is when you get it from the sex cell of one of the parents, the DNA was there, or spontaneously, which can be caused by environmental factors such as UV radiation, poison, or it can just be completely random when making new DNA. Anyways, on to the next topic of genetic mutation. Now, we're going to talk about the types of genetic mutation, and later, the causes of those genetic mutations. Okay, kids, so the first type of genetic mutation we are going to discuss is a point mutation. Okay, so point mutation is when one of the DNA bases is replaced with another. This type of mutation only affects one or a few genes in the gene sequence, so it usually doesn't have a huge effect. An example of this would be if we had a GAG coding, right? This is the normal one. And then, due to mutation, it turns into GTG. And we see that A, adenine, was replaced with T, thymine. The next type of mutation we will discuss is a frame shift mutation. Okay, so a frame shift mutation is caused due to insertion or deletion of a DNA base. When a frame shift mutation occurs, this puts off the whole DNA sequence, because like up here, it is read in pairs of threes. So it would be GAG, and then another GAG, and then another GAG. But now, if you insert or delete a base, the pattern gets messed up completely. Here's an example. So right now, it would be read as GAG, GAG. But if we, say, added a C, which is cytosine somewhere, now it would be read as GAG, CGA, with an extra G on the end. Okay, kids, does everyone have this?
Yes, Mom.
Okay. So, we have two more types of mutations to talk about. One is a nonsense mutation. And the other is a silent mutation. As we learned before, DNA is made into RNA and then a protein. So now, a nonsense mutation leads to the RNA becoming a stop codon. Okay, so now that we learned that RNA becomes a stop codon during nonsense mutation, you need to know what a stop codon is. So, a stop codon is a nucleotide triplet within the messenger RNA that signals a termination of translation. And as we learned before, translation is when RNA becomes protein. So, this type of mutation would lead to no proteins being made, which is really bad for the human body. So, a silent mutation is a mutation that doesn't change any of the physical or observable characteristics of the organism in which they occur. Take down this definition. Okay, kids, there's only a few minutes left of class, so quickly we're going to go over the causes of a few of these mutations, namely point. Okay, mutation is caused by the substitution of a DNA base. These substitutions include transition. This is a swap between adenine and guanine, also known as purines. Okay. It can also be a swap between thymine and cytosine. Also known as the pyrimidines. Okay. Do you guys all have this? Take a second to write it down, and I'll give you time to write it down before I erase the board. Okay, kids, now that you've all taken this down, we can talk about the other cause of a point mutation. Okay, so the other cause of point mutation is transversion. Transversion is a swap between one of the purines and one of the pyrimidines. Okay, kids, take that down. An example of this would be A swapping with T, or G swapping with C. Or it could be A with C and G with T. You know, either one, as long as it's a purine and a pyrimidine swapping places where they're supposed to be. Okay, take a second to take that down before we move on to the next one. All right, kids. Now that everyone got transversion, we can move on to the last cause of point mutation, which is mispairing. Also known as mismatching. Okay, so mispairing is when a purine, such as A, pairs with a pyrimidine, such as C. The correct way for bases to pair up would be A and T, or G and C. But a mispairing or mismatching would look something like A and C, or G and T. It could also be if C pairs with T and G pairs with A. You may be asking, what does she mean by pairing? So, when DNA is made into RNA, it would start as adenine, right? But during mispairing, it would become cytosine instead of thymine. That's what they mean by pairing. Alright kids, you got this? Finally, we're going to talk about large-scale mutations. Large-scale mutations are caused by translocation or inversion. Okay, so a large-scale mutation is a mutation that occurs on a chromosomal level. So it happens in your chromosomes. Translocation is when a gene from one chromosome is swapped for a gene on a different chromosome. Remember that translocation only occurs within non-homogeneous chromosomes, which is meaning they aren't the same type of chromosome. Okay, so now, inversion is when genes on the same chromosome swap their places. Okay, remember that small-scale mutations affect DNA at the base pairs, or nucleotide level, and that large-scale mutations affect DNA at the chromosomal level. I hope you all paid attention because we're going to be having a quiz on Wednesday.
The next day.
Here, Savannah. Here's the quiz.
That was so easy.
3 weeks later.
Since you girls are still here after class, do your quizzes.
Let's see what I got. A 50? What did you get, Emily?
I got 100. Oh, I have one more question. What's a mutation? Seriously?
It is all mutation changing, yes it's changing animals. Mistakes occurring in chromosomes or in genes, that's so. If it happens in a